Good afternoon, everyone, and welcome to the Infectious Disease Epidemiology Seminar Series brought to you by the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health. I am Loretta Marcosti, and I'll be your moderator for this session. I'm excited and honored to welcome Professor Jessica Metcalf as our speaker today, who is at Princeton University. First, let me introduce our speaker. Professor Jessica Metcalf is an associate professor in ecology, evolution, and public affairs at Princeton University. Professor Metcalf holds a PhD from Imperial College on the evolutionary demography of monocarpic perennials and has been with Princeton University since 2014. Professor Metcalf is a demographer with broad interests in evolutionary ecology, infectious disease dynamics, and public policy. She works on an array of health questions with a particular focus on vaccination policy, combining public health surveillance data with digital data, including mobile phone call data records and mechanistic models of infectious disease spread to characterize the intersection between human demography, behavior, and infectious disease. Professor Metcalf's talk today is entitled The Evolutionary Ecology of Coronaviruses. Once again, please remember that you can send questions during the talk to Bradford Taylor via the chat function. Thank you very much for joining us, Professor Metcalf. Please take it away.
Oh, well, thanks so much, Loretta, for that introduction, and thanks so much for inviting me. You can see my screen, right, Loretta?
Yep, we can see.
Perfect. So I think it is clear that pathogens are part of our ecology, just as we are part of theirs. And if you were alive and conscious on the planet in 2020, it was pretty hard to ignore this fact. From remarkably early on during the outbreak, we knew vast amounts about the pandemic virus, thanks to the incredible efforts of many people, in fact, many people whose names I recognize from the participants on this call. And such information sets the stage for answering, many of the current burningly urgent questions around the evolution of variants, but these remain frustratingly hard to answer. So what I'm going to do is step back and ask really broad questions about the evolutionary ecology of this virus, rather than the specific questions about what happens next. And the three questions I'm going to talk about in this talk are one which was posed by my very brilliant postdoc, Ben Rice, early on during the pandemic, which is why are there only 4 endemic coronaviruses? And I'll explain why that's mysterious. My friend and colleague Justin Lessler asked, why has MERS-CoV failed, where SARS-CoV-2 seems to have succeeded in becoming a pandemic virus? And then in work with Henrik Salge, we asked, why does COVID-19 affect people differently? And these are all, at some scale or another, really root questions in evolutionary ecology. If we consider infectious diseases in an era of global change, as in a paper led by my postdoc Rachel Baker, pathogen emergence and reemergence has been with us, for ages and over the last 20 years as well. This shows numbers of deaths on a log scale against some of these sort of classic pathogens. And clearly the SARS-CoV-2 scale is far too low because of what's happened in the time since this paper was published. So disease emergence is not surprising per se, but if we narrow in on the coronaviruses, and only the coronaviruses, there is something a bit surprising. So there have been 3 of these in the last 20 years, SARS-CoV around 2003, MERS-CoV in 2013, and then of course SARS-CoV-2. But if you think about it, that's an awful lot of coronaviruses to spill over in the last 20 years and only end up with 4 endemic coronaviruses. So the endemic pool contains those slightly, hard to remember names like HCOV229E, etc. But if you imagine that the same rate of spillover had been happening in the last 20 years, in the last 100 years, as has happened in the last 20 years, then we should have at least 15 endemic coronaviruses, but we only have 4. So why is that? Why have we got such a small endemic pool given such a vast potential number of spillover events? Of course, so you can work through a number of ways in which this mismatch might occur. And the first might simply be that we just can't count. So there might be many more endemic coronaviruses than we think. We think there are 4, but there's actually 15, 100. There's reason to think that's not the case. So a classic trick in ecology, if you want to know how good you've done at keeping track of counting things, is to just plot these accumulation curves where you plot the number of species you've counted as a function of time, and this thing should saturate at some stage because you end up running out of things to count. And even in 2012, a paper by Woolhouse et al. Suggested that this viral discovery curve, so these are viruses discovered over time, seems to be flattening. So it suggests that there's not a vast number, it's not impossible, but there's not a vast number of coronaviruses out there that we're just missing. Another piece of this that makes us think that we possibly haven't just miscounted is that if anything, we think that the bias would go the other way. Emergent pathogens, so like SARS-CoV, MERS-CoV, should, if anything, be rare and ephemeral rather than, ubiquitous and solid in the data. So we might overestimate endemics relative to emergence, but not necessarily the other way around. So it seems like the counting might be solid. So what else might be going on? It's possible, of course, that the pace of spillover has really accelerated. So I was assuming that this rate had stayed the same for the last 100 years. If instead the rate has massively increased, this pace of spillover could have increased. We could end up with many more spillovers in recent years than we would suspect if to just end up with 4. And there's reasons to think that spillover might be accelerating. So for example, there's accelerating intensity of anthropogenic disruptions to natural systems, which is very much a feature of our planet these days. But on the other hand, zoonoses do go back hundreds of years, the 18 influenza, but plagues as far back as the plague of Athens, and they predate these sort of industrial extractive processes. So this might be part of the story, but it might not be the whole of the story. There's that piece of it, but the other piece of it is, of course, if it isn't so much what we're doing in situ, it's how fast we move things around. So this shows travel connections in 1933, again from that paper on an era of global change. And in 2020, the rate of connections, these are airline connections, are just massively, massively bigger. So if something emerges in one part of the world, these days, it's going to get everywhere extremely quickly. So again, this is likely to have played a role. But it's also arguable that, the 1918 influenza did just fine long before there was this huge, massive rate of connectivity. And there are strong suggestions that a corona at least might have bubbled through in the late 1800s. So it might be that this, again, could be part of the story, but it might not be the whole story in terms of understanding why we have just the 4, despite the vast potential for spillover, of time for accumulation to occur. What else might have changed? Well, we might have changed. So what has changed in human populations, the sort of vast, flagrant change if you're a demographer, is that we're aging. And it is possible, of course, that aging populations are more propitious to spillovers. Aging individuals have senescent immune systems that just might be easier for pathogens to get into. And that might mean that's why we have more spillovers in recent years. But on the other hand, the people who are likely to be the primary exposures might be younger. So for example, people who go out and collect bats, for example, who are exposed to high risk situations are likely to be not the older individuals in populations. All right, so working through, the range of things that might be happening, it doesn't seem like the miscounting is likely to be a major issue. It could be that the rate of change of spillover has changed, but that might not be the whole story. The change in us, again, is possible, but you have to think about both change in us and change in the exposed population. So perhaps it's time to turn to think about what's going on within the pathogens themselves. And the question here is, perhaps there are limits to pathogen biology, right? So perhaps it's just very hard being a coronavirus that can successfully spread in human populations. Maybe it's very hard to get the right kind of lock and key mechanism for receptor binding. So maybe it's just vanishingly rare that happy combination, or rather a really unhappy combination, comes together. This also seems slightly unlikely, in part because coronaviruses are just incredibly diverse. There's thousands of species distributed across avian and mammalian hosts, and which suggests that they don't encounter vast barriers in their receptor ecology. Furthermore, if you look at the coronaviruses that infect humans, this is a phylogeny showing the emergent coronaviruses in red and the endemic ones in yellow, interspersed amongst some of the reservoir species coronaviruses. And you can see that they come from all over that tree. So the coronaviruses that infect humans are only distantly related. So it doesn't take something special to be able to infect humans in terms of your pathogen biology. Furthermore, this range of coronas seems to use a vast number of different receptors, right? So SARS-CoV-2 and SARS-CoV both use the ACE2 receptor, as is infamous at this stage. But MERS-CoV, the Middle Eastern Respiratory Syndrome, the coronavirus that came over in 2013, I can't remember exactly, uses a different receptor, DPP4, and some of the endemic coronaviruses use two other receptors as well. So they seem pretty promiscuous in terms of what species they can infect, what receptors they can use, how they can move between species. And this is particularly clear for SARS-CoV-2, which not only spread into our populations, but seems to be spilling back with enormous facility into just about everything. I think there was quite recently a snow leopard at San Diego Zoo, and the story about deer infections just sort of grows and becomes sort of fascinating. So it doesn't seem like the receptor binding is likely to be the massive barrier here. What else could it be? If spreading within hosts is clearly an important part of what happens for a pathogen to successfully spill over, but there's a way in which you can be too successful. So, you first get into the host, you start to replicate, and if all goes well, you're cleared by immunity. But one of the things that we have seen, of course, as part of the tragedy of this pandemic, is that you get worse symptoms associated with replication in the lower lungs, and people die. And if people die, that's not great for the pathogen, because the pathogen would rather people went on and transmitted. And it might be actually very hard for a virus to thread this needle, especially a respiratory virus with access to the lungs, because the lung is just a single cell wall thick. And if anything goes wrong, you die just really, really quickly. So it's a very delicate place to be messing around in. And so it just might be very hard for the pathogen, any pathogen, to thread that needle in terms of managing transmission and virulence if you are a respiratory pathogen. Of course, the host pathogen ecology, one of the big nexuses of those two interactions is immunity. And one can imagine that, that any pathogen trying to slip into human populations will be vulnerable to both aspects of the immune system. The first, which is innate immunity, which is where it's the part of the immune system that goes looking for kind of conserved patterns in pathogens. So parts of the pathogen protein sort of bank that cannot be changed easily. So they end up being stuck in a particular shape. And that shape is kind of straightforward for the immune system to recognize. And of course, pathogens don't want to be recognized. And so they often evolve towards looking more like the host, or as much like the host as they can. And it might be that that also is actually quite tricky for a pathogen to achieve, and tricky for it to achieve on reasonable timescales. So it might be that the kind of those classic conserved markers that are conserved for a reason are just hard for the pathogen to move around. It might also come to bear. It might also, the pathogen might be such that it also comes to the attention of the adaptive immune system simply because of its resemblance to its close relatives. So the adaptive immune system works to learn about pathogen pieces that it hasn't seen before and be ready to zap them. This is, I'm sure, sort of extraordinarily simplified way of thinking about this to almost everyone on this call, but it helps me. And there is lots of evidence, increasing evidence, that there is some sort of cross protection between the different coronaviruses. This is a rather old paper looking at the dynamics of 3 of the endemic coronaviruses in Scotland. And even just looking at this raw data, you can sort of see that in years when the orange coronavirus is big, the purple coronavirus tends to be small, which is at least suggestive of this level of cross protection. If you've been exposed to one, you're less vulnerable to the other. And you can see that this will be a challenge for something that looks like any of these things that's trying to spill into the population. We can imagine a kind of immune space in which there are already a couple of existing endemic coronaviruses, and the new guys trying to come in, it's only going to be able to do it if it can slip in the space between the cross protection from immunity that has developed to the endemic coronaviruses. So, this mystery of why there are only 4 endemic coronaviruses, it emerges from the fact that it just seems like a very small number to accumulate over human history, if you consider the fact that we've had 3 spillovers in 2 decades. beyond reporting biases, or maybe something recent that's changed massively, it seems like the answer must lie at this intersection between transmission, virulence, and existing immune cross protection. So in a way, to think about it, this reason that this number might be so small is that any new pathogen species must thread the needle in terms of managing virulence and transmission while successfully evading existing immunity in the human population. So that's the first part of the story that I was going to tackle today. I will shift now onto asking a kind of related and different angle on the story, which is thinking about why has MERS-CoV failed where SARS-CoV-2 succeeded? Okay, so we have these 4 endemic coronaviruses that are circulating happily. We've had 3 emergent ones, but they've emerged and very different things have happened. So SARS-CoV was contained and controlled. MERS-CoV seems to happen to routinely spill over, but nothing really bad has happened yet, whereas SARS-CoV-2, as we all know, rather changed the world. So what do we know about MERS-CoV? So Middle Eastern Respiratory Syndrome coronavirus. It's a mild infection in camels. It has high seroprevalence in adult camels, which suggests that it's transmitting very efficiently. There's quite a short time to the most recent common ancestor, which suggests, and this and other lines of evidence suggest that adaptation to camels might be ongoing. So they're susceptible in young, but it seems to sort of maybe this virus is still finding the right way to be a great camel pathogen. The reason that we care about this is that more than 2,000 cases have been recorded in humans, originating from multiple spillover effects. There are frequent short trains of transmission, but no large outbreaks apart from one in a hospital setting in South Korea. So it seems to sort of keep having a go at adapting to human populations, but it doesn't seem to somehow get anywhere, which is quite mysterious and interesting, especially since it has this basic capacity to do it, right? It does seem to be able to do some human-to-human transmission. So why no SARS-CoV, why no MERS-CoV pandemic? Could it be something about the reservoir? Is there something curious? And I mean, the reason we want to sort of tackle this question is, of course, we'd like to think about what are the circumstances that create super problematic pandemic potential viruses. So might it be features of the reservoir? Is there something very different about MERS-CoV that comes from camels versus SARS-CoV or SARS-CoV-2 that comes from some intricate mix-up of bats and civet cats and so on? Well, there do seem to be ways in which the reservoir matters. So if you look at the ratio within the genome of MERS-CoV of Cs followed by Gs, so this is called CpG, then you find that MERS-CoV, as shown on this plot here from a paper by Nichua et al., is rather high. So Cs followed by Gs are quite common in the MERS genotype, a genome, whereas for SARS-CoV-2, it's rather low. And this matters because the human immune system, and the innate immune system in particular, has a suite of tools to rapidly zap anything that has a very high CpG ratio. And this is because our own CpG ratios are very low for reasons that have to do with methylation, not fundamental to the story, but it means that a very straightforward marker of an alien, an intrusive and problematic alien, is a very low CpG ratio. Sorry, very high CpG ratio. And it seems like when SARS-CoV-2 came into human populations, it had a very low CpG ratio. And it's been suggested, I think in that paper, that circulation in bats might have just fortuitously selected for lower CpG and thus innate immune evasion in SARS-CoV-2. So SARS-CoV-2 can fly under the radar of that particular part of the immune system just because it got lucky, whereas it seems like MERS-CoV did not get lucky. Nothing about camels predisposes you to have this low ratio. So features of the reservoir might have shaped the vulnerability of MERS-CoV to the innate immune system. There's also potentially some things about the ecology of the zoonotic reservoir that shake out in terms of how vulnerable MERS-CoV is to the adaptive immune system. So quite a lot of work has shown, well, not a lot of work. There's been a very limited amount of work in many parts of Africa in trying to understand what MERS-CoV circulation is, but there's increasingly some lovely papers have come out. And it was suggested in a study of abattoir workers, camel abattoir workers, that it seems that individuals who worked in these jobs that are very, very closely connected to camels and camel slaughter, etc., had T-cell immunity, elevated T-cell immunity to MERS-CoV. So quite surprising, because they'd never shown symptoms and they didn't have antibodies, but they might be, simply because they are in these sort of close forward-facing exposure positions, they might be creating an immune wall by having developed adaptive immunity to this camel. There's also more complexity to the story there. It has been suggested that there might be milder strains circulating in Africa as well. But again, this is another way in which MERS-CoV's ecology and zoonotic reservoir is really rather different and creates these circumstances where you could build up this adaptive immunity that would prevent it from spreading further. So zoonotic reservoir could matter. It could be also, of course, once you've got out of the zoonotic reservoir and into the host, there's going to be differences between SARS-CoV-2 and SARS-CoV, in fact, and MERS-CoVs within host growth. And there's also been some very lovely comparative work there, lots of it involving Alan Perelson. So this is a paper by Kim et al., which looked at the time courses of the within host growth of these three emergent coronaviruses. And you can use exactly the same descriptors we use for spread of infections at the population scale to understand how infections are spreading within hosts. So like cell to cell R0 can be estimated. And when they did that, they found that MERS-CoV seemed to have a very low R0 relative to SARS-CoV-2 and SARS-CoV in terms of how it spread inside the lung. And of course, this summary number encompasses just a vast amount of underlying mechanism and ecology. So you've got to take into account that, there's going to be differences in viral cell entry, there's going to be differences in replication within cells, there's differences between innate and adaptive immunity that I alluded to. All these different pieces are going to come into play in shaping how this different within host trajectory for SARS-CoV-2 and MERS shakes out. And I realize I never described this graph, so let me just tell you what it is quickly. On the x-axis is days after symptom onset, and red shows SARS-CoV-2, so coming up super quickly and then falling away. In green is SARS-CoV, so that first coronavirus that emerged, coming up pretty quickly, but not as, not in the Olympic stakes of SARS-CoV-2. And then MERS comes up very gradually and kind of dies away. So these things, these inherent differences may come into play as to why MERS-CoV can't actually make it up to that pandemic scale. Another piece of it, which relates again to thinking about the why only for endemic coronaviruses, is to do with the specific biology of how the receptor works. So I told you earlier that MERS-CoV uses a different receptor from SARS-CoV and SARS-CoV-2, and it uses this receptor called the DPP4 receptor. And the DPP4 receptor has a distribution that's like this in camels. So it goes from very low in the lower respiratory tract to very high in the upper respiratory tract, which is perfect for a virus that just wants to transmit a lot and would rather not kill its host. Not that viruses necessarily want to do that, but in this particular case. But that receptor, who knows exactly why, has a different geography in us. So it has a low density high up and a pretty high density low down. And if you're using that receptor, that means for MERS-CoV, you really can't avoid being deep in the lungs, or at least it's probably quite hard to avoid being deep in the lungs, which is the part that results in really high mortality. So it might be extremely hard for MERS-CoV to kind of evade being a massively virulent pathogen, which would to the detriment of its own transmission. Of course, I mean, the coronavirus ecology and evolution is just absolutely fascinating. So there is an exemplar that makes the point that this is not, this doesn't necessarily have to be endgame for MERS-CoV. There's a pig coronavirus, which transmissible gastroenteritis coronavirus that was very virulent in pigs and then seemed to disappear, although they were still pulling out in 1984, they were identifying animals with tons of antibodies still to this virus, even though they didn't seem to be getting sick from what they thought was this pathogen. And it turns out there'd been changes in the spike receptor, and it seems to have turned into a respiratory coronavirus that was very mild. So there does seem to be ways in which at least cellular tropism can massively change within the coronaviruses in ways that might, will change the ecology and the transmission. So it's not that being stuck with a DPP4 receptor will completely stymie the future of MERS-CoV in terms of being a pathogen of humans, but it might be another barrier. So we talked about the features of the zoonotic reservoir, features of how these things grow up inside us. And then, of course, one has to consider features of transmission process, so how these things move from one individual to another. It's been apparent since the lovely papers of Christoph Fraser and work from many people at Harvard and beyond, that features of the transmission process, in particular timing relative to symptoms, are really going to be critical for whether a pathogen can take off or not. And looking at these three curves for the different emergent coronaviruses, one can see that SARS-CoV-2, just by screaming up to really high levels very quickly, may be able to getting most of its transmission done before symptoms are apparent, and this is indeed the case. But we just have a very sort of crappy understanding of how you map from viremia to symptoms to all these different processes, so it's hard to sort of pin down what mechanisms might be at play there that we really have to think about that might be a barrier to MERS-CoV becoming a pandemic virus, beyond the fact that it's moving slowly, it's not getting up very high. Another thing about when you move from one individual to another, if you are a coronavirus or indeed any virus, in lovely work by Cachiquela and others has probed this for SARS-CoV-2, is that although we have millions of viruses inside us, only a few move from one individual to another. And so there's really strong bottlenecks in this process. And so this leads to sort of another barrier to evolving from moving within individuals to between individuals. And in particular, if you had any misalignment between what it took to grow up inside an individual and what it takes to spread between individuals, say survive in the air or move efficiently from one upper respiratory tract to another, if those things go in different directions, if what it takes to be good at one is not what it takes to be good at another, then the efficiency of spread of those mutations will be even more diminished by the fact that you have this very severe bottleneck. And it seems, again, for SARS-CoV-2 has this, unfortunately for us and fortuitously for it, this seems to have this strong alignment between things that are good for how it spreads within us to how it spreads between us. So, a greater stickiness to the receptor, the ACE2 receptor, helps within host growth and seems to help between host growth as well. This might not be the case for MERS-CoV, which might be one of those things that traps it. All of these coronaviruses seem to, very weirdly, to have very super spreadery type dynamics, and super spreadery type dynamics amplify your chances of extinction. And this means that you end up again with like a much reduced potential for your variants that are super adaptive and super efficient to spread through the population. So, I think that the exercise of thinking through what means that MERS-CoV doesn't seem to be able to make it, whereas SARS-CoV-2 did, one of the things that really brought home to us is that, while we often have a tendency to think about the singular event, the index case or the critical mutation, there's much more, there's so much going on in terms of what it takes to be a successful emergent pathogen, that you need to be considering this range of features. And a theory of viral evolution is needed. And people like Jamie Lloyd-Smith have been saying this for years, but it's always good to convince oneself of things over again. So you need to bridge that gap between viral genomics and viral epidemiology. And just to put it in super abstract terms, again, we can think about crunching it down to just sort of epically too simple. If you think about mutational space on the x-axis here, then fitness on the y-axis, where you have fitness in a reservoir host, and fitness in humans, where the key number, of course, for emergence is to have R0 greater than 1. And you can imagine that whatever the situation, variants in the reservoir are going to be concentrated, the ones that exist are going to be concentrated around the higher fitness levels, just because we expect adaptation to occur, selection to drive adaptation, etc., if these things are genetically possible. And if you imagine that where the population is focused will describe the range that can be reached by mutations, in this situation, it might look like this, in which case there's no virus that can come out of the reservoir that will successfully invade humans, right? So this mutation has a fitness of 0 in a human, even if it's right up at the boundaries of the mutational range of what's possible in the reservoir. If instead those two distributions are much closer, then you can imagine something up against the limits, up against the edges of the mutation range, could spread within human populations. And if you imagine that range, the kind of mutational range that's in the reservoir, say it's a really complicated multi-host reservoir of bats, you might end up with a much broader mutational range. So something at the margins of the mutational range of the reservoir could actually result in vast fitness, so it could come in basically pre-adapted to spreading in humans. And of course, we really want to understand, we want to move beyond these schematics to sort of think about what specific things could we be measuring to understand whether there are a ton of viruses out there that are pre-adaptive, and the real concern is that we'll bump into them, or whether there are a ton of viruses out there that are close to being adapted, but might come into our populations and then through stuttering chains of transmission, find a way through, or the things that don't matter at all. And I think one of the sort of things about thinking about in very concrete terms about what differentiates MERS-CoV from SARS-CoV-2 is it begins to give us a sense of what it might be interesting to measure. So for example, and I would never have thought about this before, it's presumably pretty straightforward to develop an atlas of what the distribution of the different receptors are across our respiratory tracts. Are they ones that are at much higher densities, lower versus higher? That it feels like one should be able to do that across a range of hosts in a relatively straightforward manner. There's also the interesting question of if, we've contrasted the ACE2 receptor versus the DPP4 receptor, is there a way in which ACE2, just all mammalian ACE2s look a lot like each other? So if you can do one, you can do them all, versus DPP4, maybe they're much more distinguished. Is there a way in which we can make a mapping of the range of these different receptors and how tractable it might be if you were a virus if you could use one to use all of them? I think that this is and from conversely, if you start going out and sequencing a bunch of viruses, you should again be able to understand the range, the degree to which, and people have started doing this. There's some lovely papers out there looking at what it would take, how much you have to tweak a virus spike protein in a coronavirus to be able to enable it to invade mammals with a range of different, the same receptor, but across a range of different mammals. This is a very spike-centric set of things that one might measure, and of course, spike is not all. So I think another set of really interesting, very clearly mechanistic processes that one could start grappling with is things like the CpG ratio, right? So these kind of clear tells that we know something about a very sort of gross property of the immune system, and one could go out and look across the virome and see if we could identify things that seem to be in that sort of worrying spot. There is, of course, the vast interest in seeing this happen. The SARS-CoV-2 has spilled over into deer. Longitudinal data on what happens next and how that process has moved on. Of course, we might be a little bit late for the deer, but can we catch other items where they start going on so we can understand that ongoing adaptation to a new host, and therefore start to identify what the flashpoints, what the danger signs might be. And of course, I do think that adaptive immunity is likely to be at least part of the story in what creates a fire shield against these pandemic potential pathogens. And so having a bit of a landscape of immunity in terms of measurement of antibodies might be a powerful way of, again, just laying out where the risks lie and starting to understand the underlying evolutionary ecology of pandemic viruses. So those two pieces move us, or at least helped me think at least, that I was beginning to get a sense of what that landscape of pandemic risk pathogens might look like. There's another question, sort of another way of framing the questions, which is, which are the ones that are going to hurt us? Which are the ones that are most pernicious to our health? And a way to frame this question, an alternate framing for this question, is why does COVID-19 affect people differently? So this is just the piece I'll go over in the last third of this talk. And of course, the reason that SARS-CoV-2 is such a pernicious pathogen is because it combines this capacity for invisible spread in some individuals with extremely severe, extremely negative outcomes in others. There are some very mysterious parts of this as well. Early on in the pandemic, it seemed so clear that individuals who are otherwise identical could suffer extremely different outcomes in the context of the virus. And we speculated early on that this might be illuminated by thinking about the underlying evolutionary ecology of the interception between the immune system and viral growth, because immune feedbacks open the way to alternative stable states. And so you can sort of maybe start thinking about how this might underpin cytokine storms. But beyond that, these vastly divergent outcomes in otherwise identical individuals, there were also clear tells of individuals who are more at risk. And this shows data from France looking at the number of hospitalizations for males and females across age. And of course, we know now sort of enormously that the clearest risk factor for SARS-CoV-2, mortality, hospitalization, is age. So individuals at higher ages are at much higher risk. And we also know that there are sex differences in the risk. Okay, so what might underpin this? What shapes these differences in our outcomes? Is there a way in which we can think about this by taking a few steps back into evolutionary ecology? And it turns out that evolutionary ecology thinks very deeply about why senescence has evolved at all. And I'll just walk you through the mystery and its resolution. So the mystery comes from the question of, if you're thinking of everything as wanting to maximize fitness, you would imagine that everything should try and live forever and have as many babies as possible, right? In very, very crude terms. And of course, that's not what we see. So why isn't that what we see? And so people like Medawar and Williams, back at the dawn of evolutionary biology, not the dawn because that was Darwin, but a little later on, the midday, identified a really sort of crisp, simple reason that this was the fact, which I will now illustrate. So if you imagine that you have a population of 2,000 individuals, and you imagine you're in a very difficult situation, or you're in a very particular species where every year the risk of dying is 50%. So that means the cohort is 2,000 individuals are born. So the cohort has 2,000 individuals at age 1. At age 2, you have 1,000 individuals. At age 3, you have 500 individuals. At age 4, you have 250, and so on. Okay, so this is what your demographic profile will look like, because your probability of dying is 50%. Okay, now imagine that a mutation pops up in this population that increases the mortality of 2-year-olds and increases it by quite a lot. The impact is because there's going to be considerably fewer 3-year-olds, considerably fewer 4-year-olds, and considerably fewer 5-year-olds, and so on. So you end up with this rather big decrease in the population as a result of this one age increase in mortality. Okay. Now imagine that instead of occurring at age 2, an exactly identical mutation occurs at age 8. Nothing happens. I mean, something happens formally, because exactly, if you're in an infinite population, that fraction would die, but there's no discernible effect simply because there are so few individuals left alive at that age, right? And so the result is that mutations that increase late life mortality are expected to increase. You expect this to occur either because something that they do to increase survival or fertility early in life leads to this cost you pay late in life, but it has so little effect that you actually end up with spread of those early benefits. Or it could be just there's nobody there. So these mutations that increase mortality can drift to fixation. So if you're never going to live to age 60, or all your children are never going to live to age 60, if you pass on a mutation that results in mortality at age 60, it's not going to affect them necessarily before they have their children, and so it can spread in the population. So we sort of absolutely expect increases in mortality at late ages from the context of background evolutionary ecology. What does this mean in terms of thinking about SARS-CoV-2 and thinking about the burden of this particular infection? Well, we can lay out our expectations for what's happening in SARS-CoV-2 by comparing it to other sources of mortality. So setting it in the context of other ways in which we die, and setting in the context of thinking about what the baseline is, which should be the outcome of this longer term process. This again is work with Henrik Salge and lots of wonderful people at Public Health France. So what I'm going to do is I'm going to show you relative risks across age on the x-axis of hospitalization here, but we also looked at other outcomes. And I'm going to start by focusing, and I'm going to anchor every single one of the things I compare at age 60. So they're going to have a relative risk of 1 at age 60, just so as to have them all on the same scale. So let's start with influenza, all-cause mortality, and the endemic coronaviruses that we were thinking about in that first question. So this is, you can see all 3 of those things seem to have a very similar, like a strikingly similar pattern on hospitalization. So hospitalization is very high if you're very young, then it kind of flattens out, and then it increases at late ages. And that late age increase is that pattern of senescence at old age. This is known amongst demographers as the bathtub shape of mortality, very characteristic. Okay, and again, anchored at age 60 because we're trying to compare the overall shapes. Now what happens if we throw in a couple of our emergent coronaviruses? Well, they look like this. So they map very closely, that late age senescence pattern seems to look a lot like what we see in the other things, but the difference is happening at much younger ages. So you do get this decline in mortality at the left end of the scale, but it goes down much deeper and sort of comes out of a much later, as a sort of a much, it starts increasing, that relative risk starts increasing somewhere in the range of puberty. So it seems likely, based on the comparison with influenza and the coronaviruses, that this difference is due to adaptive immunity. So the thing that kind of softens out and creates that plateau in the bathtub is likely to be the fact that individuals are at risk early in childhood, then they develop immune memory to these different pathogens, and then it kind of flattens it out until senescence kicks in and they start dying again. And thinking about it as an evolutionary ecologist, this is actually very weird. And it's very weird because the adaptive immune system is very weird. The adaptive immune system does something which no other physiological process, well, apart from brains, does, which is learn. It sort of learns what's going on, and it allows things that kill you early in life can actually have a protective, or put you at risk early in life, a protective later in life. So an early cost can be a benefit, and this shifts the calculus a little bit for the evolution of senescence. And I think, it makes the point as well that all pathogens are going to be emergent in some sense for children. They're necessarily experiencing them for the first time. And this was presumably a responsive innate immunity in early life allows that first learning that then yields later protection. This might be the baseline for those sorts of infectious diseases, which also emphasizes a kind of third point here, which is that the strangest thing about SARS-CoV-2, in some sense, was that none of us had been exposed to it before. So it really was that learning piece, that flattening out in the middle that seems to have been missing. Okay, so that's what happens if we stare at the differences over age. What happens if we stare at the differences over sex? So this shows you patterns of females above the line and males below the line in terms of number of hospitalizations. And it might also already be apparent to you that they do seem to be differences in the nuances of those patterns. So I'm going to do the same thing just to sort of pull this out a little bit more. I'm going to plot the relative risk for the different sources of mortality, but now I'm going to plot it with excess male risk above the line and excess female risk below the line. And what I'm going to do to aid comparison is I'm going to anchor relative risk at 20 to be 1. And so to start off, here's all-cause mortality. This might not be something you know. I think it's kind of weird and interesting. So males die more. This is not death. Are at greater risk of hospitalization across all ages, but similar patterns are seen for mortality. And there's very high mortality, and then it sort of goes down to around 20, and then it starts increasing later on. And the very late ages, the fact that you see a decline again in the relative risk in males might be something to do with selection effect. It's actually quite hard to tease apart the different processes at play there. Okay, so that's all causes. What do we see when we throw on the infectious diseases? So the infectious diseases, I'm sorry, there's a bit of a spaghetti plot, but they look like this. So all three of the groups here, so it's SARS-CoV-2 in red, influenza in blue, and the endemic coronaviruses as the thin black line, show that same pattern of higher male mortality early on, then it declines and then it goes up. But what we're seeing is that during sort of between 20, so during the middle years of life, women are actually at a higher risk of hospitalization, and we also see this in mortality, than men. So certainly older men are more at risk, but within those middle years, women seem to be more at risk. And this is really sort of interesting and peculiar, this tipping between the different ages, where there's excess male risk early in life, this tilt towards female risk in the middle of life, and then back to excess male risk, which seems to be sort of so striking and so sort of consistent to some degree across these different infections, is, I think, very surprising. One can come up with reasons as to how this works and what the sort of driving processes are. But I think at this stage in the talk, we're not going to go down that rabbit hole. I'm just going to quote a lovely paper by Sabra Klein, which makes the point that we know strikingly little about sex differences in immunity. And this is something that I think I could talk about for hours and I find very fascinating. And I'll just put up the peacock to make that comparison clear. And so I think there's lots of really interesting things to work on and think about there in terms of those underlying mechanisms as well. But just to summarize, what I've tried to do in this talk is talk you through how we can sort of bound these problems about thinking about what leads, how the evolutionary ecology perspective raises these questions which might seem a little surprising, sort of reveals these surprises that we can use to think about what the problems are, and then thinking about our own side as well. And I just want to mention a lot of these ideas were driven by my postdoc Benjamin Rice, who's particularly on those first two, and then Justin Lessler and Henrik Salje, who worked with me on the last piece, and the last piece also includes a lot of lovely collaborators from Santé Publique France and other organizations, and Derek Cummings and Simon Cachemez. So thank you very much.
Thank you, Professor Metcalf. That was a wonderful talk. So we'll now take questions, and we'll try our best to accommodate as many questions as we can. However, if your question doesn't get covered, please feel free to reach out to our speaker directly. Also remember that you can still submit your questions via the chat to Bradford. So our first question is from Larissa Nealon, who asks, I feel like part of the MERS story was that it was too virulent for widespread transmission. Is this part of the story or too oversimplified?
So I think that definitely is a large part of the story. But there is also some proportion of cases which are asymptomatic, or at least weakly symptomatic. So I think it's 20%, which isn't vast, obviously. It does seem to be an incredibly virulent pathogen. But I think also I think that certainly that one barrier could be enough to block you entirely. But it feels like, you could compensate for that in other ways if you had different levels of transmission, et cetera. And that's sort of part of what we were trying to do in thinking through this.
And our next question is from Catriona Shea, and she'd like to ask the question herself. If she could unmute.
Hi, thank you. That was a really interesting talk, and I'm sure everybody is concerned about like more virulent strains. And so when I first learned evolutionary ecology a long time ago, the idea was that you became more transmissible and less virulent, and that's kind of a good general rule. But obviously with the sort of asymptomatic stages that you were talking about, it's perfectly possible for a variant that is is asymptomatic for a month and then fatal. And that would be highly effective, even if it didn't have very high transmissibility. And we've seen with coronavirus, with SARS-CoV-2, with the variants, some that have been more virulent and some that have been less virulent if you measure it in terms of fatality or hospitalization. Do you have a sense for what we can expect? Sorry, that was a long preamble, just to ask that question.
My brilliant graduate student, Ian Miller, has written a paper on thinking about how we might expect virulence evolution to play out in the context of vaccination. And a point that he makes very crisply in that paper is for virulence evolution in general, virulence by itself is just bad. So virulence needs to be correlated with transmission for it to have any effect at all. And then whether you get an increase or a decrease will depend on which side of that optimization hump you are in terms of virulence, right? You might be too virulent, in which case you'd decline. You might be not virulent enough, in which case you'd increase. But I think you laid it out perfectly, Katrina, right? The link between virulence and transmission in SARS-CoV-2 is pretty nebulous. And it's pretty nebulous in part because much of the mortality happens long after transmission, or at least not long after transmission, but to some degree after transmission. And you could still believe that, that the things that stop transmission were actually morbidity. People stop moving around, so that could reduce transmission. It's not impossible that there is a link between the two, but based on what we've seen so far, it's very hard to say what that mapping would be. And, Omicron, we wrote the paper before Omicron, then Omicron, and it was all very confusing. But Omicron suggests it can do both. It can do whatever it likes. But it could also be that, evolutionary ecologists classically think in terms of these trade-off bands. And it could just be far away. It seems to be that it's very far away still from the edges of those trade-offs. So it's still traveling up towards them.
Yeah, thank you for that. And our next question is from Jonathan Dushoff. I think he'd like to ask a question himself.
Hello. Apparently I haven't connected before and I'm getting yelled at by my browser. Hi. Thanks for the talk. I was just wondering, have you tried those cool anchored plots you showed with age, but particularly with sex? Have you tried plotting the same thing without the anchor? Because I was imagining in my brain different possible scenarios for the sex thing, and I didn't know if you'd had the chance to look at that.
I think we did do that, and I can't remember exactly what it looked like. I think in part, one always worries in that of differences in healthcare seeking behavior, right? But of course that could play out over age as well, so that it doesn't save you to anchor things in particular. Yeah, you're absolutely. I don't have those pictures in memory. There was presumably a reason we went for those anchored plots, but
Thanks.
And our next question is from Sang-Yuk Shin, who asks, is the sex difference pattern for hospitalization conditioned on infection? Asking because exposure to infection differs due to social factors, not immunological.
This is another great question, and I think also links to what I was answering to Jonathan. I mean, there's many reasons we might expect there to be differences in risk and differences in risky behaviors, well, as alongside differences in health-seeking behavior in men and women. And one of the challenges we've had throughout this pandemic, of course, is we just don't know what the denominators are of the people who turn up, who are tested, who turn up in hospital. So that's certainly, it wasn't conditional on infection because we just didn't have that data, because I think, no one really has that data except possibly the ONS in the UK, which is not going to be funded anymore. So yeah, that would have been wonderful, but we just didn't have that information.
Yeah. And our next question is from Ben Balka, and he'd like to ask it himself.
Hi there.
Hey, Ben.
Round up the usual suspects. I was curious, in the graph that you showed of viral load over time in MERS, and the reference, and I guess the original paper was about differences in R0 among those. But what's sort of obvious from that graph is differences in timescale, in the rate of growth, which might, and I assume that it does balance out in the way that R0 is turns out to be less. But I wondered if and you also mentioned that the timing of symptom onset is important. And I wondered here if maybe the timing of symptom onset and therefore the effective window of transmission was more important than the actual rate of cell-to-cell spread.
It's extremely astutely and concisely put, as expected, Ben. No, I think that's absolutely right. And I don't, presumably there is data that one would allow one to start picking at that for SARS-CoV-2, I guess maybe not for MERS-CoV or for SARS-CoV. But certainly, there are some, there has been a raft of work out of those groups thinking about heterogeneity and spread within the lungs. All the things that we worry about when we throw out a single number R0 come into play when you start thinking about the actual sort of intricacies of what it means to move around a lung, what it means to think about susceptible cells, right? Because if an interferon is splashing around, et cetera, et cetera. So there is it's certainly an awful lot of nuance in how those timescales, how that susceptible depletion plays out, whether it's to do with immunity or not. So yeah, absolutely great point. And
so it's complicated. Thank you.
That's what I'm saying.
Yeah. Our next question is from Bradford, and he'd like to ask himself.
Awesome, thank you. Yeah, great talk. Yeah, so my question was kind of related to the spillover and just the idea of the mutational space in which you can then move over from one to the other. Now, obviously, we're looking for it, but I feel like I've just anecdotally heard a lot about reverse zoonoses, with the deer, mink, and whatever else. So I'm wondering if you could like maybe just speculate on conceptually the difference between sort of spilling over from say from a bat to a human and then from a human elsewhere, and whether or not you would expect there to be like vast differences for any number of reasons why maybe there'd be more spillover from humans, like more readily available reverse zoonoses than vice versa, or perhaps it's a sampling issue.
I think this is, I wish I knew the answer to this question, right, Bradford? Because
speculate, no. Yeah.
But I think this is one of the things it would be just great to have a handle on. I'm not sure. I find that thinking about mutational space, crunching it down to fitness landscapes helps me think about what's going on, but it might be very misleading, right? We're taking a whole mass of traits and putting them down into one place. And so, for example, some of Cachiquela's work, which looks at how flu might end up in these neutral spaces, and you need to travel around a lot before you end up in somewhere where you can do these massive jumps. So it might not be that Euclidean representation might not be representative at all. But I think what I was trying to get at is that by trying to think very specifically about very specific mechanisms, we might find ways to start bounding those processes. So for example, on the CPG front, on the receptor distribution front, and maybe, the people have speculated that bats are special, bats are weird because they have, weird interferon, et cetera, Could we answer that? Could we get to go out there and like measure some of these core parameters, or measure an ecosystem that we understand well, and figure out what the circulating viruses are and how they're moving between things. I also think, just on the last point, we tend to, which I think you were hinting at as well, like we are the vast reservoir for everything else. We're the source of infections, we're the origin of TB, we are the problem host for most things. So that's certainly worth bearing in mind.
Thank you.
On behalf of the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health, let's give a big thank you to Professor Metcalf again for joining us today and for a fantastic talk and discussion. Please do get in touch with any of the seminar organizers if you have questions for us or our speaker. Also check out the Center for Communicable Disease Dynamics website to stay up to date on upcoming seminars and other events. We will have a break next week, and the seminar series will resume on March 24th. The next seminar will be given by Professor James Collins from MIT, and he'll be talking about harnessing deep learning and synthetic biology to fight pathogens. Thank you again for joining, everyone, and goodbye.